Hello everyone, welcome to the ITLS Academy. Okay, before starting anything, do you know what is DNA? Okay, let me tell you. DNA is a deoxyribonucleic acid, which is the genetic material of ours, basically human genetic material, and it is hereditary, which can be transferred from our parents to our offspring. So we can say from generation to generation, due to which we can see variations in us. Okay. So today we are here to perform the isolation or extraction of DNA from human saliva. Okay, for that we required these are the material which we required for performing like micropipette reagent. So this is the material which we required for performing isolation of DNA from human saliva. This is Eppendorf, and this is our Tris EDTA buffer, which act as a buffer solution. This is micropipette, and these are test tube holders, and water obviously for which we need to rinse. So first of all, for the very first step is we mark on the Eppendorf. You can see here I mark the first word of my name. This is T. So first of all we need to mark the Eppendorf. Secondly, what we need to do, we need to take a sip of water and we need to rinse it up to 10 minutes. So this is the rinse water after 10 minutes. Now what we need to do, we need to transfer 1.5 microliter of this solution into every Eppendorf. We set the micropipette on 500 and now we are going to take the solution and transfer it into each Eppendorf. By the same procedure, we need to transfer the solution into each Eppendorf. Now we already transfer the solution into each Eppendorf. First, now we are going to perform centrifugation, and we need to perform centrifuge for the 2 minutes at full speed. Now you need to keep all these into the centrifuge. You need to make sure that you can put these Eppendorf in the balance. See, as you see. Okay. Time 2 minutes at the full speed. Now start this. See, now you can see the time and it is full speed right now. So after 2 minutes we need to take out all these Eppendorf. Now after this we need to discard the supernatant and we need to perform the procedure in the same manner for at least 2-3 times for getting the better results. Okay. See, now what we are going to do, we are going to discard the supernatant. And remember, we need to after discarding the supernatant, further we need to centrifuge the Eppendorf in the centrifugation, and we need to do it at least 2-3 times for getting the better result. Now after discarding all the supernatant, you can see the pellets here. we need to centrifuge it again for at least for the same time period.